Hey there. Hey there, Psych 9 students. Professor Zeeb here. Welcome to your next audio-visual lecture. In this short lecture, we're going to talk about brain development, some different facts about the brain, and even ways that we actually research it in a class like this in development and psychology and so forth. All right, so let's go to our slides here and check it out. We'll go over the lecture here. All right, so let's talk about some brain development. It turns out that brain development actually starts very early on. So if we look at our different stages in prenatal development, it actually begins in the embryonic period. In fact, during this time, we're going to find that the top of the neural tube leads to the formation of the two hemispheres. So the brain consists of two hemispheres, a left and a right, and actually has four major lobes, which we'll talk about in just a second. So just to kind of give you an indication here, as little early on as 3 weeks, we can see the three major parts of the brain forming. Those are called the forebrain, the midbrain, and the hindbrain. So 3 weeks in after conception, we can actually see those forming. 7 weeks in, still technically in the embryonic period, we see the forebrain, hindbrain, midbrain, and so forth sort of forming. And then 11 weeks, when we're technically a fetus, we really are seeing those being further defined. And then of course at birth, there you go. So just some general points here. Keep in mind that at birth, the brain is about half a pound or about 25% developed, which means a lot of that development does happen in the womb, but even more so after we are born, especially the first 2 years, as we're going to find out. Just to give you an indication, by the end of the first year, we would expect that brain to go from half a pound to about a pound and a half on average. So it shows that there's massive, massive growth rates during that time. The brain is what we call localized. So it's very localized, which basically means that specific brain parts control specific functions. Now, guys, you can relax for the exam. I'm not going to make you memorize all these different things here on this particular slide, but just for demonstration purposes, here we have what's called the temporal lobe. So as you can see, that's responsible for things like hearing, advanced visual processing, where the frontal lobe, a very important, excuse me, part of the lobe, our brain, is movement, working memory, decision-making, et cetera, et cetera. So the point is that the brain has specific regions that control specific functions, okay? And if that particular part of the brain was compromised, well, then we would see some damaging effects in those areas. So for example, the back of the brain is called the occipital lobe, excuse me, and that's where we process visual information. So if you're like in a car accident or something happened, we know people who have gotten this part of the brain damaged actually could go blind. So the point is that those specific regions control those specific functions, and thus the brain is extremely localized. This next slide shows our basic cell of the nervous system, which we call a neuron. There are many types of neurons with different shapes and functions, but this is our general structure. So a neuron has three basic parts. We have our cell body that you see here, which contains the nucleus. We have dendrites, which are these branching structures, and they receive information. And then we have like a tail section, which is called the axon, which helps to send messages. So the way the nervous system works, and of course the brain, is it uses electrical chemical impulses to send information throughout the body via the neurons, okay? So in a sense, neurons are kind of like messengers. For example, if you stubbed your toe, that information would travel up your leg, up your spine to your brain, where you would sense the pain of stubbing your toe. So the neurons enable those messages to be sent throughout the body, as well as let the body complete different types of functions, okay? Once again, they do that by using electrical chemical impulses, right? So in other words, an exchange of chemicals and creating a charge to distribute those messages throughout the body. All right, so next we go to neurons and brain growth. Based on our studies and research, we have discovered that brain cells can be replaced. You know, for the longest time, they thought that once brain cells were damaged, that they were irreplaceable. Now we are finding that that is quite not the case. In fact, research shows that brain cells can migrate and attach to the damaged areas and then transform into appropriate type cells. So for example, if you had a stroke, which obviously creates extreme stress on the brain, that person may lose certain abilities immediately following the stroke because their neurons have been damaged. We have found that over time, the person can regain some of those functions to a certain extent because the neurons are basically repairing themselves. So we've found that they can actually regenerate and repair. So now we've come to a really important part of this particular chapter in the class in general. And it's this sort of phrase that we use them or lose them. Research basically shows that if neurons are not stimulated, they can die. See, it turns out that we are born with way more neurons than we actually need. And so we have found that the brain goes through this natural pruning process over time. Excuse me. So the point is that if your brain is not being stimulated, some of those losses would be greater. So it's so important, guys, with your child to be stimulating their mind constantly to prevent some of these losses and also to strengthen those different parts of the brain. In other words, it's really showing the importance of stimulation from the environment. As infants absorb information from their environment, we have found that the brain grows. So it actually grows and weakens depending on their experiences. Furthermore, in this particular study by Bick et al. 2017, found that depressed brain activity has been connected to growing up in impoverished environments. So theoretically, if we kind of understand that, it means that children who grow up in these environments that are not stimulating and are lacking certain resources, for example, we have seen that it depresses brain activity and thus growth, and potentially some of their neurons would perish and die as a result of that. So the bottom line is, guys, as we're all becoming parents or are parents, it's very important to stimulate your child's mind constantly to incorporate faster growth and strengthening of those connections between the neurons. For example, you want to read to them, you want to talk to them, even if they don't have language yet. You know, play music for them is something I did with my own children. Show them different objects, encourage movement. All of those types of activities causes the neurons to basically function and to grow and make stronger connections among themselves in these networks of neural connections. So very, very important. So as the very famous John Lennon once said, I love this particular quote, he basically stated that your brain is like a muscle, it needs exercise. I love that. Obviously the brain is not actually a muscle, but you kind of see the point here. Just like our muscles need exercise to become strong and to grow and things like that, the brain's kind of the same thing. We should be actively stimulating our brain. And by the way, that's not just children, that's all the way through life. We should be doing that constantly, and we'll examine that more in later chapters. But because the brain is really being laid down during infancy, especially that first year, it's very important to be stimulating your child's mind to foster growth and to encourage development. Keep this in mind, our genetics basically provide the basic wiring of our brain and the neural circuits, if you would, but experience and environment are guiding the development of the brain. So once again, it's the classic nature nurture stuff that we've been talking about that we laid down early in class. Both of those are very important, and so very important to stimulate your child's mind. Okay, so next we're gonna switch gears here, and we're actually gonna go over different ways that we research the brain. So we're gonna look at some different tools that psychologists and scientists use to examine the brain. I mean, this is one of the ways we know what we know in terms of how the brain functions. So let's go over our different strategies. Excuse me, the first one is called the electroencephalogram, or an EEG as it's known. This particular device detects electrical impulses of the neurons. Remember, we said that they use electrical chemical impulses. And so as you can see here in the photograph, these electrodes would be placed on the scalp. And so they monitor brain activity. More specifically, they're going to measure neuron activity and help to identify different behavioral states and look at how the brain changes during those times. So for example, sleep, when we're aroused, for example, it's going to monitor the brain waves based on neural activity to give us a reading. So that is hooked up to a computer to allow us to sort of examine that. So that's called the EEG. The next one we're going to talk about is called a CAT scan. Computed axial tomography is the fancy nerdy term there. With this particular technique, we're using X-rays to take cross-sectional pictures or slices, if you will, of the brain. So this one's kind of neat because it produces a 3D picture of the brain. And then those images or the slices are combined to kind of give an overall picture of the brain, which allows us to assess what's going on. Okay, so next we go to the PET scan, more technically called the positron emission tomography. So this one, it's a little bit complicated, but essentially what's happening is we're injecting chemicals into the person's bloodstream first, and then these are detected by a computer. In other words, we're injecting these radioactive isotopes into the bloodstream, which act as a tracer that the computer can actually detect when they're being examined. So let me kind of show you how these sort of work. So because those chemicals, those tracers are being injected into the bloodstream, we know that that bloodstream, of course, travels to the brain, and theoretically, a brain structure that is receiving more blood flow technically would be more active. So it allows us to monitor that and as someone is experiencing something. So let's look at some of these photographs here. So for example, if you look over here on the left, this one says visual. In this particular example, someone is looking at an image or something, and you'll notice that the back part of the brain has this red orangish color. So the way you interpret these is that the warmer colors, red, orange, show more blood flow and therefore more activity, where our cooler colors, like you see up here, blue, show less blood flow and less activity. So going back to this particular picture, this shows that the person is looking at something and that this part of the brain is very active when they're doing so. And remember, we said at the back of the brain is the occipital lobe where we process visual information. So this makes complete sense. This part, the visual part of the brain, is being activated when the person is experiencing a visual image. So these types of studies, these types of machines allow us to see what's happening during different events or different environmental stimulation. So for example, music or language, and notice how the readings are a little bit different for each one. So it's allowed psychologists and scientists to identify which brain structures are connected to different activities and different experiences, if you will. All right, so next we go to the MRI, magnetic resonance imaging. This particular technique uses magnetic detectors to measure hemoglobin in the blood, or basically oxygen levels. So it's going to measure the internal structures of the brain and shows us potential damage. So it actually creates a more detailed image than, say, the CAT scan, and allows us to look for lesions or problems with the brain. And it also measures blood flow within the brain, okay? So it's sort of an expensive piece of equipment, and it's very limiting because the person typically would lie on their back and go in the machine. And so psychologists are kind of concerned about this one because obviously the person's in a very static condition when they're being examined by this. So now we have technology that kind of gets around that. And let's go to our next one here. It's called the functional near-infrared spectroscopy. And I'm probably mispronouncing that, but you can see it there up on the screen. And so this one is going to use low levels of near-infrared light to monitor changes in blood oxygen. But the advantage here is that it allows the baby or child to be able to move and to explore and not just lie on their back in a machine. So you can see here in the photograph, they've attached these electrodes to their brain, and there's wires connected to the machine, of course, but allows them to actually move around and explore the environment, which gives us more rich detail in terms of what's happening in the brain during these various activities compared to what we were able to do before in a very static kind of environment. So we think that moving disrupts a lot of the other scans that we've talked about. So this has kind of overcome some of those obstacles to allow the child to move and not be in a completely sedentary type condition. All right, so that will do it for this particular short lecture. Hopefully I've helped you understand a little bit more about the brain and how we actually study it. Thank you for listening to the lecture, and we'll see you in class or online.